use an immunobiobank that we generated here at the College of Veterinary Medicine that is capable of trying to predict vaccine efficacy or vaccine immunogenicity to a specific per strain that we are currently isolating from North Carolina swine herd. The project is very focused on North Carolina pork industry, and we are trying to help veterinarian in giving them some tool to reduce the burden of this relevant disease in the field.
Welcome to the Swine Canada podcast show, brought to you by Wisenetics. We are dedicated to bringing you the latest insights and discussions in the Canadian pork industry. So my name is Miranda Smith, and I'll be the host for today's episode. And today's guest is Dr. Elisa Crisci, who is an assistant professor at the North Carolina State University. So welcome to the show, Elisa.
Thank you. I'm very happy to be here with you.
Yeah, so today we're going to be talking about some of the research you're doing, and specifically a project on the PreProPRRSV project, which is quite a mouthful. But before we get into that, can you first give us a little bit of a background or an introduction to your background and the role you're currently doing?
Sure. So I've joined North Carolina State University in 2018 as an assistant professor. And my main expertise is in swine virology and immunology. And I've been working on this topic for practically 20 years now. That has been always my main interest, everything that is related to viruses and pig viral diseases. And in particular, I've always been very interested in virus-host interactions and focusing also on immunity and, for example, vaccine responses, everything that is related to immune responses. So since I started, I built up my lab working on swine viral infection, in particular respiratory infection. And we started mainly with two viruses. One is influenza virus and one is PRRSV, or porcine respiratory and reproductive syndrome virus. Both are important and relevant viral diseases in the industry. Right now, in particular for PRRS, they have estimated about 1.2 billion per year of economic loss only in US. On the other side, influenza is a relevant pig virus, but also a relevant public health concern because it's a zoonotic virus. So my lab has been working around these two viruses for both agriculture perspective, but also using pig as a biomedical model for example, influenza studies. So we've been working on this for 7.5 years right now.
Swine Veterinary Partners comprises 4 well-established clinics across Canada: Precision Veterinary Services, Premier SHP, Demeter, Ontario, and Demeter, Quebec.
Okay, so it sounds like you're doing very important research. So the last 7.5 years, what are some of the main things that you've looked at?
Yeah, from the perspective of PRRS, we have evaluated in particular a lot of virus-host interaction and pathogenesis studies. And more recently, we have moved more towards vaccine testing or, for example, efficacy studies. And the latest one that we have actually is ongoing right now that is called PreProPRRS is an interdisciplinary project that is funded by USDA NIFA IDEAS program, and combine different expertise to try to solve this PRRS puzzle, I say. So one side has an epidemiology and a modeling portion that is led by Dr. Gustavo Machado, and one portion is what I'm working on mainly, that is the virology and immunology portion. The name is pre-pro because we have a proactive and prediction model. So the prediction model is actually the one that we do with the modeling, and then we have a proactive system try to use an immunobiobank that we generated here at the College of Veterinary Medicine that is capable of trying to predict vaccine efficacy or vaccine immunogenicity towards specific PRRS strain that we are currently isolating from North Carolina swine herd. The project is very focused on North Carolina industry, pork industry, and we are trying to help veterinarian in giving them some tool to reduce the burden of this relevant disease in the field. I can go more into detail in the project if you want to.
Sure, but before we do that, maybe just a little bit of background on the PRRS virus for people like me who are not as familiar with it. So, it sounds like there's a lot of different types out there, and that's why it's hard to create vaccines. Is that right?
That's correct. So, PRRS is a RNA virus, and actually within the RNA virus has one of the highest rate of mutation. Because of that, there are continuous emerging and re-emerging strain worldwide. I mean, this is not an issue just located in the North America. We actually have two main species of PRRS. We have what they call European or type 1 and what they call the North American or type 2. Here we mainly have the presence of the PRRS type 2, some level of PRRS type 1, but very, very limited. Because of the high rate mutation, this virus is able to mutate a lot, but also recombined, for example, with strain that we use for vaccines. And that's, of course, created an additional problem in the capacity of control this disease with the tool we have. Currently, there are 5 available modified live vaccines in US that are commercially available in the market. They are all modified live vaccines, so we have an attenuated strain of PRRS that is used to vaccinate the animals. One of the issues with this vaccination is that they are pretty effective when the strain that is in the vaccine match the one that is in the field. But because of the high rate of mutation, when there is a mismatch between the vaccine strain and the one that is circulating, they tend to not have a very high level of protection in the field. So there is some limited cross-reactivity or cross-protection between, the strain that are included in the vaccine and the one that are actually circulating in the field.
Okay, and so that's where the Biobank comes in?
Exactly. So what we're trying to do with our commercial available vaccine, of course, we would like to have a vaccine every single time we have a new strain, but of course you understand that is pretty difficult to implement in the field. What we are doing is we have created a biobank that is practically contain PBMCs, so cells and plasma, mainly to target antibodies that are actually coming from vaccinated pigs that have been vaccinated with the 5 different commercially available vaccines. What we do, we use an in vitro system that is based on seroneutralization assay or ELISpot assay that try to predict how immunogenic is that vaccines with a specific strain that we have actually, for example, isolated from the field maybe 6 months ago or 2 months ago. So we are trying to isolate the most recent strain that are circulating in North Carolina and try to test those strains in our biobank and see if this in vitro system that is based on what we call correlated protection can in some way give an idea which vaccine will be the best shot in trying to protect the farm, for example, towards specific strain that is circulating. So we are trying to give some tool to improve, the capacity to contain an outbreak of PRRS when that happens in the field.
So how many strains have you tested so far?
Yes, so far we have tested 6 North Carolina strain. We have also used what we call prototype strain. Those are mainly Midwest strain that have been considered prototype for a very long time. And so we have tested all of them, and we have actually seen different profile for each vaccine group, depending on which strain we have actually been using. We have combined that with in both cellular immunity and seroneutralization that measure neutralizing antibodies. Unfortunately, the time point that we have collected these samples was not really targeted for neutralizing antibodies. That tends to be pretty late in terms of the vaccines tend to induce pretty late neutralizing antibodies, generally after a month. So we did our biobank by collecting the sample after 1 month of vaccination. So we were literally at the kind of limit of starting to see some neutralizing antibodies. But on the other side, we choose that time point because it was one of the best for cellular immunity. And that's why our early spot is providing a lot of information about how the different vaccines behave towards a specific strain that we are testing.
So can you explain a bit more on like some of the results?
Yeah, sure. So for example, we have seen that with a specific, strain that has a specific classification. I just want to clarify, PRRS classification have been evolving through the years. At the beginning, we were always using in the veterinary diagnostic lab NOR5 sequencing that was based on what we call RFLP, so a specific section of the OR5. Based on that classification, strain would have been called 174, 173, 134, 184, 144. So there was some type of classification for specific strain that were isolated. Now the classification of per strain has been evolving to more lineage and sublineage and variant type, still based on ORF5 sequencing, but with a different classification. So most of these strains now are mainly, for example, included in what we call lineage 1A. That is one of the most frequent lineage present here in North Carolina. Other strains, for example, L1C, are now circulating in the Midwest. So what we have been doing and finding out that there will some difference in vaccine response depending if, for example, the strain isolated was, included in what we call lineage 1A or lineage 1C. And for example, some vaccines, I can name the different vaccine, but right now I'm not sure if it's that really of interest. But some vaccines were behaving much better with a specific strain, for example, isolated last year, because they were giving much more interferon gamma producing cells response in our in vitro system. So it's practically we are basing our prediction on the level of interferon gamma that these PBMCs are actually producing in vitro.
So can farmers use that biobank already, or are we still more at the research stage here?
We already been using it. We actually had given some recommendation to some veterinarians in the field. Of course, this is the first step. So we are establishing the biobank. And there is also kind of a plan to improve the biobank in the future. So we have planned a pig trial for next year where we are trying to expand a little bit more the time point. And we are trying to, for example, include a challenge of the pigs too, to see if we can combine in vivo data. So let's say the result that we have of protection or not in a challenge trial, and compare those in vivo data with our in vitro data that we use to estimate the efficacy of a specific vaccine. So that will help us give a much more correlation between what we really see in vivo and what we predict in vitro.
Yeah, that makes a lot of sense, because what happens in a test tube does not necessarily happen out in the field.
Yeah, exactly. We also have to consider, and you probably everyone understand, we cannot make a challenge trial for every single strain that we isolate from the field, in particular with a virus like this one that mutates so much. So we are trying to optimize our resources. So for example, the commercial vaccines that we are currently have in the market, with the best correlated protection and in vitro system that we can develop, in a lab setting. Of course, the best would be to have a vaccine specific for each strain, but this is clearly not feasible. So we are trying to find the best way to predict this kind of immunogenicity with an in vitro system.
And so at what point would you say the PRRS virus has recombined or changed enough that a new vaccine would be developed?
This is a good question. Unfortunately, most of the vaccines we currently have in the market are kind of old, in the sense that we do not have vaccines that, for example, have been developed in the last 5 years on strains that have been kind of circulating in the last 5 years. There are even vaccines that have even more than 25 years of kind of since they've been produced. Yes. So there is, of course, some limitation because it's kind of hard for the industry to keep up in terms of, creating new vaccines that match circulating strain. So the best would be to find a vaccines or a vaccination system that will cover all strains. But unfortunately, right now, this is still not a reality. I really hope it will be, but unfortunately, there are a lot of scientists and researchers working on new vaccines approach, new platform. Still, we have limitation with the type of kind of tools that we have against PRRS. There is also a lot of work done on biosafety measure and everything else that is not just vaccines. And I'm sure probably some people have heard already, we have new genetic modified pigs that might actually be resistant for PRRS infection. And recently, FDA has approved these genetic modified pigs. I don't know if this will be the future of the industry. There is a new approach that we can use against this very detrimental disease. But right now, what we currently have available are mainly vaccines approach and biosafety measure approaches.
Yeah, like the new pig sounds really interesting. So it'll be interesting to see if that becomes like the main type of pig that we're using in the commercial industry, or if we're still far away from that. But
unfortunately, it's a question I don't have an answer.
Yeah.
And not sure if anyone has an answer on that. But at least one step has been, done towards a much more resistant pig. And I cannot say more than this.
Yeah, fair enough. So you were mentioning for the Biobank, it's currently mostly focused on North Carolina. Is there a hope that it will be expanded to national or international?
Yeah, so the idea of the Biobank needed to start it on a kind of local level, also for us to be able to combine that with our prediction system that is actually, for example, modeling how vaccination in farms decrease or modulate the spread of the viruses. Dr. Machado has access actually to most of the North Carolina farms and information on those farms. He has also access to other farms that are out of North Carolina. So, of course, the initial idea of this project was to start local, and then after we the proof of principle, after we prove that this system actually can help the industry, the idea would be to expand it to, for example, to the Midwest. Regarding the international, the concept can be used in other areas, of course, but we have to think that, for example, European strains are different than, North Carolina. So PRRS type 1 is different from PRRS type 2. So there will be, for sure, a need to establish a biobank that is based on PRRS type 1 in Europe, for example. if this concept is actually going to be beneficial, and we see that it's going to help the industry to reduce the burden of the disease in the field.
Yeah, that makes sense. Okay, so is there anything else around the PreProPRRSV project that we haven't discussed yet?
Not really. We are actually going to be soon releasing the preprint of this first biobank, so stay tuned on online release of our article that I hope it will be of interest for all the swine community and swine researcher. The future of this project is to, as I said, to create a new biobank with a little bit more defined approach and refine targeting different time points, and of course trying to use in parallel a challenge to compare in vivo with in vitro. So that will be the last step of this big project, and we really hope that at the end of this big project we will have more information, and we will have a kind of useful tool for the pig industry, in particular in US.
Okay, perfect. Well, I'm looking forward to that. So
thank you.
It's time for our famous three.
Okay, well, thank you so much. So now we're going to the final three questions, which is always the same for all our guests. So the first question that I have is, what is your favorite swine-related book or resource?
I found very hard to answer this question because I have so many sources. But I would say because I'm located in North Carolina, one of the main resources I generally look at is SHIC, so the Swine Health Information Center, that provide different information on report on circulating disease, in particularly viral diseases in the pig production in US. Then, of course, we have what we call the Bible, the Disease of Swine book. We have several swine podcasts and a lot of social media that actually provide information on swine industry. So I think we have a pretty big a portfolio of social media and other resources that we have available for swine research, and I'm very happy about that. So yeah, that's for sure. Maybe I didn't answer with one answer, but there are so many that is really difficult to choose between all of those.
Yeah, that's fair. And it's a good thing to have so many different options. So yes. Okay, so then the next question is, what is your favorite book or resource outside of agriculture?
This will maybe people laugh, but recently I've been reading a lot of books on kids. I have a 4.5-year-old son, so I'm reading a lot of information to try to be able to understand his behaviors and try to keep up as a mom on how he's, his kind of behavior, and trying to understand just how kids' brain works. So this might be funny, but that's currently the non-scientific, if we can say, related book that I'm currently reading. The other one is the immunobiology, but that's completely, related to my expertise. So I'm biased in that sense.
Well, I think it makes a lot of sense to read books on kids, because I don't have kids myself, but I often think raising kids is one of the hardest jobs, and it doesn't come with a standard operating procedure. So
yes, I was not trained in that, unfortunately.
Yeah, we don't have PhDs in raising kids. So okay, and then the last question. So in your opinion, what sets successful swine professionals apart from those that are not?
My personal opinion in this is, I think those what I found the most, inspiring people and swine researcher are often the ones that have a great communication skills, people that can really communicate at any level, anything that they want. And so I found that very helpful in a leadership role. The other kind of thing I think is important is how good collaborators they are. So I found that also very relevant throughout my career, to find people that are very approachable, and they are available in helping you, for example, trying to understand, research question, or helping you in research question, or even just try to answer practical questions that are currently ongoing in the industry, or even in the research setting. So I think communication and collaboration skills are, at least for me, the two very important skills that I found out to be very relevant in leadership, not just in the swine industry or swine research related field, but in general. I think in the research realm and in the veterinary realm, as I'm a veterinarian, so in just different disciplines.
Yeah, that makes sense. I think the like swine leadership is not all that different from other kind of leadership. So we're probably looking for similar things in different leaders, right? So okay, perfect. So thank you so much for joining me on the Swine in Canada podcast show today.
Thank you very much for the invitation. I was very honored to be invited for this podcast. Thank you.
Excellent. So to our listeners, thanks everyone for joining us today. So don't forget to subscribe to our podcast to stay updated on future episodes. And if you enjoyed today's episode, please leave us a review, because your feedback is invaluable to us. So that concludes another episode of the Swine in Canada podcast show. So until next time.